Andreas is always great and handsome man.
So, can you see my presentation,
Yes,
Ahmed?
yes, yes,
Yes.
yes.
Yes. So, of course, I would also like to thank the organizers very much for inviting me to this well-known, in the meantime, well-known conference, which is held almost always at the same time in November. Okay. And it's a great pleasure for me to attend again.
Thank you, Dr. Andreas.
Yeah?
I would like just to introduce you to the
Oh,
audience.
I'm sorry.
Before you start. No problem.
Please.
Also, it's my pleasure to have Dr. Andreas Börner in the IPGG for the third time. Dr. Andreas Börner is like a fixed speaker in the first one and second one, and now he is with us in the third IPGG conference. Dr. Andreas Börner got his PhD in plant breeding and plant genetics from Martin Luther University. Then he was head of the research group with genetics of the IPGG Institute. He joined the Genebank of the IPGG as the head of research group resources, genetics and reproduction in 1997. He has served as the head of program management and evaluation. In addition, his responsibility for the optimization of management of the collection, he runs an active research program focused on the investigation of seed longevity, the study of genetic integrity and genetic diversity, as well as comprehensive phenotypic and genetic description of material related to the major temperate cereal crops, in close cooperation with many external partners. Segregation, situation mapping, population are exploited to detect genes QTL responsible for agronomic characters of the interest. Thank you, Dr. Andreas, for joining us, and please go ahead to start your presentation.
Yes, thank you again, Ahmed, for introducing me and for giving me the chance to talk about the crop germplasm. And we have heard from Rajiv a great presentation about all these genomic tools which are available now. And but we should always keep in mind we also need the material to work on and to use these tools. And Rajiv did show us that he has genotypes 3,000 lines and so on. But from where comes this material? This material is mainly stored in our gene banks, and therefore I would like to give you first some introduction into the gene bank work, and in fact I will divide my presentation into 4 parts. So I will talk about plant genetic resources on a global scale, what's going on our globe with respect to storing plant genetic resources. Of course, I will say a few words about the German ex situ collection at the IPGG in Gartersleben, and then the main part is I will give examples on research. And as Ahmed already told in his introduction, we are doing a lot of work on seed longevity. That means how long a seed does survive, because in order to work with the plants, we need living seeds. I will give some example on stress tolerance. We did research, and I will give some conclusions and outlook. But let me start with a global view, and I have shown this slide already several times, but nevertheless, I think it's very good to start. What you see here is our globe, and we have 7.4 million accessions worldwide stored in about 1,750 gene banks. And the 10 largest gene banks here I have highlighted with these light blue columns. And these are the gene banks in the US, the gene bank in Mexico. There is this International Wheat and Maize Breeding Institute, the CIMMYT, as most of you know. Then there is the IPGG gene bank under the top 10. This is also important. We have the gene bank in Russia, in St. Petersburg, which was also the first gene bank in the world. Maybe you know the famous researcher Vavilov, who did initiate all this work more than 100 years ago. There was the Icarda gene bank in Aleppo, but as all of you know, they had to move to Morocco and to Lebanon because of this political problems they had. We have the national gene banks in India, China, South Korea, and Japan. And we have this red column here, and this is a special storage place at the so-called Svalbard Islands. This is an international seed store where the gene banks from all over the world have the chance to send their seeds for safety duplication. If you look for the numbers, again, you see that so far the largest gene bank is in the US with about 600,000 accessions, followed by India, China, Russia, Japan, Mexico, Icarda, and the IPK in Gartersleben. And if we look for the crops which are stored in our international gene banks, it becomes clear that the two crops feeding the world, wheat and rice, also having the highest numbers of accessions in our worldwide banks, followed by barley, maize, phaseolus bean, sorghum, soybean, oat, groundnut, and cotton. So these are the big crops held in our banks worldwide. And this brings me already to the German ex situ collection, because I come from Germany, so I have to say some words about our collection as well. And some of you know already this very nice campus here of the IPK, where the institute is placed, and the gene bank department is one of 4 scientific departments, which is placed rather in the middle of the institute. You see this rather modern building. These are our cold store facilities. And then you see these many small glass houses and also bigger ones we are using for the regeneration, because we have to regenerate a certain amount of accessions each year. And especially these small glass houses we need for the regeneration of cross-pollinating species. You have to isolate them, okay? And here in the back of this picture, you see some part of our experimental fields, or here on the right-hand side, where we are also doing regeneration of the material. Back to the numbers. So we have 150,000 accessions in our bank. With this, we are the largest bank within the European Union and under the top 10 worldwide, as I already mentioned. And as on a global scale, the cereals and the grasses having the highest numbers. And within this group, you see wheat, barley, or oats, so just a few are mentioned: legumes, vegetables, oil fiber crops, medicinal spice plants, and so on. And these 150,000 accessions belong to more than 3,000 species and nearly 800 genera. And with that, we are also one of the most diverse gene banks in the world, beside the bank in the US. And we also have a huge collection or reference collections with more than 400,000 herbarium sheets, as you see here on this photograph, 100,000 samples of seeds and fruits, and 52,000 samples of cereal spikes. And we use this, as it is mentioned here, as for reference reasons, but we are also able, and we did quite some experiments, to get DNA from these old samples and to compare them, for example, with the material which is nowadays in our bank, and also for other reasons. So this is also possible. And I should mention that in 2008 we have implemented a so-called quality management system. That means all processes going on in the bank are well documented, that people know exactly how to regenerate which species, how to store the species, and so on. And I will bring one example later on in my presentation as well. The material goes back to collection missions. The oldest material comes from the 1920s. And you see here, there were areas like Hindu Kush, Tibet, China, Iran, Mongolia, Cuba, North Korea, Ethiopia. It's impossible anymore to go to such places to collect material, But at that time, in the 1960s, 70s, people were not so much sensitive for genetic resources, and therefore the material in this bank has a very high value as well. Here is one of these examples of the quality management systems we are using. This is one of the so-called working instructions for seed storage. It is in German. Of course, you cannot read, but I will explain how we store the seeds in our bank. So we have a so-called active sample, which is stored as -18 degrees Celsius in such glass jars in huge cold chambers. And then we have a basic sample, which is stored in aluminum bags under vacuum, also at -18 degrees Celsius. And this sample stays in the cold storage, whereas the active sample we take out seeds for distribution, we put them back, and so on. And last not least, there is the safety duplicates I mentioned in the beginning, which are sent to the Svalbard Islands. They are also in such aluminum bags at -18 degrees Celsius. And here you see where these islands are in the north of Norway. There is a so-called permafrost, and in the frozen rocks, they build such chambers where the seeds are stored. And 2 years ago, I had the chance to go with 2 of my colleagues to visit this place, and there were about -30 degrees Celsius. It was not so easy to survive, but anyway, it was a very interesting experience. We did send so far nearly 60,000 samples from our bank to this place for safety storage, and this is about 40% of the whole collection. And why it's not the complete collection yet? Because you should know that our cold store facility started in the 1970s, and we have now material which is stored there for more than 40 years. And we were thinking it makes no sense to send material which has already been stored for such a long time to the Svalbard seed store. So we only send fresh harvested material to this place. That means it will need maybe another 15 years before the completed collection is there, but then we are sure that this is always fresh materials. Of course, we have to do reproduction. We have each year between 6,000 and 8,000 accessions in the field or in the greenhouses. We have so-called in vivo maintenance of 1,500 vegetatively propagated accessions, and we provide during this regeneration work material for the herbarium collection. Distribution of the material, very important, because we are also distributing about 15,000 to 20,000 samples each year to all over the world. This is not always the case. I know many gene banks from where it is very hard to get any material, but in our case we did not make any differences due to political systems or whatever. So we are providing the material. However, the users have to sign a material transfer agreement. Yes, so based on this international document, we are providing the material to anybody who is needing or asking us for seeds. And as you see here in this graph, you become aware that during the 2014-16, the distributions went up to 50,000 each year. And this was not manageable anymore. So we had to stop to provide the material freely. And we did introduce some fund for some so-called handling fee. And handling fee means from 2017, people have to pay €2 per accession, plus €10 per shipment, plus the cost for the phytosanitary certificate. And having introduced this handling, this fee, you see that the distribution numbers went down again between 15 and 20,000, and this is what we really can do. However, not everybody have to pay this money. There are countries which are still free of charge. And if you see here, Egypt is also one of these countries. So if you order material from our bank, you still get it for free. Yes, and Rajiv also mentioned already that now whole collections, or that we are now able to genotype whole collections, and we started such a work at IPGG also with barley, with phaseolus, with tomato, and so on. And we hope that beside of, let's say, diversity studies and so on, we can also, this can bring us to some rationalization and for the identification of duplicates within the collection to make the management easier. And as you see, for example, here, these two accessions of barley, they seem to be really highly identical, but we cannot only, or we do not only use this genotypic information, we also have then to grow the material side by side and to check whether they are also from the morphological point of view very similar or the same, and then we may decide whether we exclude one of both or just put them together or whatever. This is not finally decided yet. This brings me to the research part. The presentation, seed longevity. This is always important, and I also reported something last year about it because for us it is very important to know how long we can store a seed. Yes, you cannot store the seed for an unlimited time. And so we started investigations which seeds from our gene bank but stored at ambient conditions. That means room temperature conditions. And the results you see here. And it is important, this red line, because this red line gives us the so-called P50 value. That means the germination drops down to 50%. And you see for wheat, for example, this P50 value is about 7 years, right? At room temperature. Yes, keep in mind. For rye, this is a cereal which is used in northern Eastern Europe. There we have a 6 years value. For maize, we have even 12 years. So this is a very good performer with respect to seed storage. Two other examples, Lactuca lettuce, for example, it's only 4 years, and you see after 6 years or 7 years the seed is dead. And Pisum, again, a very good performer. The P50 value is here for about 14 years. So there is a difference, there is a variation between the species, what we call an interspecific variation. The species are responding in a different way. But we see something else here in the case of Pisum. At the beginning, the variation is rather small. And later on, after 15 years or so, we have a huge variation within one species. So there is also an intraspecific variation. This is important. And so these were results at room temperature. If we look to our seed store, I already mentioned that it was built in the 1970s and the oldest material from harvest 1974. And we have historical germination data, more than 150,000. And we did analyze these data. And one example is shown here for wheat. The graph is looking different, but again, important is this red line here. This is the P50 value. And as you see, we can store wheat at minus 18 degrees Celsius for 35, 38, 40, maybe, or even 50 years without declining a decline in the germination. Barley looks very different, as you see here. So it's fine under such conditions here in these big shelves, in the big chambers. But if you look for rye, I mentioned at the beginning, this is a special cereal we use in Northern Eastern Europe. There is a difference. It's fine at the beginning, but then you see it's going down. Yes, and after 30 years, again, we have this huge variation. And you should keep in mind that this material here, these accessions, they are grown in the same year, the same field, side by side. They were harvested with the same and threshed with the same machines at the more or less the same time. and then it went to the cold storage sitting side by side in the shelves. So the differences we find after 30 years are not really due to the environmental conditions, but they are due to genetic differences. They have a genetic background or genetic reason, and therefore we start to investigate this. And in order to do this, we used so-called artificial aging methods. I have no time to go into details, but in general, you treat the seed with a high relative humidity and a high temperature. And we did a first study in barley, and we were able really to find loci responsible for the germination after controlled deterioration or after artificial aging. You see all these peaks here indicating that there is a QTL or a locus for seed longevity. We did the same study later on in wheat. And for wheat, it was interesting. We investigated this population also for dormancy. Dormancy is a period of non-germination after maturity. And the idea was if a seed has a high dormancy, maybe it's also surviving longer. But from this genetic point of view, it became clear that the loci for seed longevity were on different chromosomes compared to this one locus we found or one QTL we found for seed dormancy. So it seems not to be related to each other. We did repeat this experiment with a higher marker density quite recently and could identify the same regions, but also new regions for the traits in the same wheat population. We did a study with oilseed rape, and the interesting point here was that the same population was tested in 3 different years. And depending on the year, we found different regions or different QTLs. So this means that the growing conditions of the mother plant have a high impact on the seed survival later on. And another example, very recently, we also did some experiment with tobacco and could also find loci responsible for the seed long for this trait. So at the end, we were happy and we could publish several nice papers. It's always important to have publications, you know this. But what we really found were regions in the genome which may be responsible for the trait without knowing more about anything about the mechanisms. And therefore we started to do some so-called metabolite profiling. My colleagues did this here in the IPGG, and we used wheat as a cereal again, stored under different conditions, coming from different harvests years, so having different germination percentages, and these germination percentages were correlated with the metabolite profiling. And this was what my colleagues found. They looked for lipids and oxidized lipids in this wheat seeds, and in total they found 20,000 chromatographic features, and they were correlated with the germination. And there were about 11,000 which significantly correlated negatively or positively with the germination. And here is one example showing a very nice and very strong negative correlation. It's a so-called O1-deacylglycerol. And we hope that we can develop methods which helps us to predict or to find out the germination without doing these destructive germination tests, which are also quite time consuming. Recently we started, okay, this was published. If you're interested, you can see in this paper. And recently we started a rather new method we want to use for germination testing. It's the so-called delayed luminescence. So there is some light coming to the seed, and in a way, as it comes back, we may, or this we may correlate with the seed germination. We did some preliminary work with brassica, with rapeseed, and the results are very promising, but not ready for practical use. And this brings me to the last short part, the stress tolerance, where we also did work using Genebank material. And this is something which may be interested, especially for you, the frost tolerance in wheat. You never face with such a problem. But here in Europe, we have we may have quite hard winters and a lot of snow, and we have to look for material which survives such conditions. And here we started an experiment where we did grow material at different sites in Germany, in Hungary, in Russia, and even in Siberia, where it is very cold in the winter time. And the material was genotyped, and we did the GWAS analysis, and we found something interesting here in this special year on chromosome 1b, and the same in the following year. So here really seems to be something which is important for frost tolerance. Another example on drought tolerance, and this is the material, this is the origin of the accessions, it's not the testing places, because the material was tested in Germany. But in Germany, we do not have really drought. We may have some drought, but in some years, but not so severe to use it for phenotypic selection. So we use the special method, or we are using special method we are calling chemical desiccation. So we are spraying half of the plot with a potassium iodide solution. This is a method which has been described much earlier. And after 2 weeks, you see how the plants look like. The chlorophyll was killed here on the right-hand side, and also the seeds are shriveled. And by doing this, we are able to do then the mapping, and we found several loci here on the whole genome responsible for this drought stress, or for this response to chemical desiccation. Data have been published in 2019. We did a similar work with barley also. And here you see again the differences between control and drought treatment. And here you see the effect on the grains in barley. And again, here the differences in the 1,000 grain weight after drought treatment or after control treatment. And again, we were able to detect loci on all seven barley chromosomes. And we were able also to publish this data together with Ahmed Al-Kuda, who will be the next speaker. And finishing this part, I would like to draw your attention to this publication we did together with Ahmed Salam and Ahmed Al-Kuda, Stephen Bensinger and myself, and review drought stress tolerance in wheat and barley, advances in physiology, breeding and genetics research. So for all of you who are interested in drought, have a look at this very interesting paper. And with that, I come to the conclusions and outlook. 40 minutes are exactly over. So I hope I could show you that there is a huge amount of accessions stored in global ex situ collections. 7.4 million. The question is it enough? Do we need more? Storability, longevity of the germplasm in seed banks is limited. Yeah, you cannot store the seeds for an unlimited time. I know many gene banks. They collected material in the 1980s, 1990s. They put it just in the freezer and never touched it again. So I'm very afraid that this material is slowly dying. You need to do a frequent regeneration of the material. You need to do the test determination. So far, till we have tests which are maybe easier to handle. There's an inter- and intraspecific variation for longevity. There are genetic loci determining seed longevity, which we could detect in several crop species. Lipid oxidation and hydrolysis events seem to be responsible for the death of the seed. And plant genetic resources can be successful used for mapping loci responsible for stress tolerance. And with that, my time is over. Not 40, 30 minutes are over, and there are still 10 minutes for questions and discussions and so on. Thank you very much for your attention.
Thank you, Andreas, for very interesting and useful presentation. And now time for questions. So please raise your hand if you have any questions. Yes, we have in the chat. Yeah, okay. We'll start with. Yes, yes, Hiwan, go ahead, please.
Thank you very much for this educational and informative presentation. I'm currently working on barley. I'm located in the southern part of Ethiopia, and I am collecting different lines. And in your talk, you mentioned about the difficulty of sharing germplasm from Ethiopia and to the rest of the world. And that is really very informative. And I'm really wanted to say that I'm here willing to collaborate with Egypt and also with Germany. Actually, I am already starting collaborating with Professor Sato from Okayama University. And we are doing the genotyping and everything. But in your talk, you mentioned about the difficulty of storage and longevity of germination in what is rye? Is that rye or like an oat? So what is the problem with that? Thank you very much.
Yeah, we don't know. Rye is not oat. Rye is Zicale cereale. And we know from our experience that it is not storable well. But why, I cannot tell you yet. It needs more investigation to find out what's going on in this species. This was just our observation. And to do such observations, you need really seeds which have been stored for a long time. And we are happy that we have seeds stored for more than 40 years now. But I can't tell you any reason for this. I'm sorry.
Okay, thank you.
Okay, Andreas, we have questions in the chat room. A lot of questions, actually. Very nice presentation. What is the best way to control or resolve photosensitivity issue during seed multiplication process?
Wait, what is the best way to control or resolve photosensitivity issue during seed multiplication? Okay. Yeah, this is a problem, maybe a problem, because we have a long photo period here in Germany, and we can in the field only regenerate really material which comes from comparable regions. But sometimes we have material which needs a short photo period, and for this we have in fact two special greenhouses where we can make where we have a black cover, and we can have short days also during our normal summertime. But this is a problem, and it's very difficult to regenerate such plants under our conditions. Maybe one can find partners helping us, but yeah, I don't know really a solution.
Okay, thank you, Dr. Andreas. We have a question from Dr. Rajiv. Please unmute your mic and go ahead.
Yeah, I can read it. I have it here.
Okay,
thanks.
just he raised your hand, so I think okay, you can.
Okay.
Hello, Andreas.
Yeah.
Okay. No, I think Andreas, great presentation as usual. Very good data, nice research, nice work. Now my question is, as you showed the slide, in 2017 the distribution reached to about 50,000 samples per annum, and you mentioned it is quite challenging to distribute. I agree. And then you started to ask users to pay some fee. Then the requests have gone down. Now my issue is that the title of your presentation is the utilization of that germplasm for the crop improvement. My worry is by doing these things, and if all the Genebank start to do like that one, don't you think the request and in the turn the utilization of germplasm may go down, which is just opposite to the essence and the objective of the Genebanks? What do you say about that?
Yeah,
Thank you, Andreas.
Yeah, thank you very much for this question. Of course, there was a lot of discussion also before we made this step. And first, I must say that developing countries are still free. So this is one thing. And for the others, I think €2 per accession, it's nothing. It's not. And if you apply for projects, for example, then you apply for money for stuff, you apply for money for chemicals, and then you should think also if you want to test association panels to think about money for this material, because the gene banks, we cannot always regenerate the huge amount of seeds for nothing, you know, for free. This is not possible. And I know that the Genebank in the Netherlands, they are also thinking about some kind of system. And I also should say, when we did implement the system, I was very much afraid that I get a lot of phone calls and people saying, "Oh, what are you doing? It's impossible." No, it was not the case. People did really accept this, and you know, people start to think about why they order what. If everything is for nothing, then people say, "Oh, I take 500, also I can only work with 100. It doesn't matter. It is for free." I still believe this was the right decision to do so.
Okay, thank you. Because Germany, Netherlands, they are very rich country. They could manage this thing. But I understand the point. Thank you. Thank you.
Andreas, we come again to the chat room and we have some more questions.
Yeah.
So Gauda asks, very nice presentation. What is the best way to control? No, this is one we already told. So في Hafiz Omar Farouk,
Yeah.
I was wondering, how did you perform phenotyping for identification of the loci associated with seed longevity?
Yes.
Did you face some challenges?
No, I mean, what you need is seeds which have a certain age or which you did age artificially, and then you just do the germination of these lines, and either it is a biparental population or it is an association mapping panel, it doesn't matter. And then you do just the classical genetic study based on the data of germination. So there we divide also into total germination or plants which performing normal seedlings, or you can also, for example, measure seedling and root lengths and something like this. And these traits you correlate to your genotypic data, and then you detect the loci. But you can read it in these papers. It is described very, very clearly, and it's not really a big problem. Oh, here I see. Thank you.
Yeah.
Is mislabeling, misclassification, or taxonomy error can occur in a gene bank? How to resolve them? What are the best tools for this? Oh, yeah, this is also a very nice and complex question. Of course, there can be always mistakes. Each regeneration step may lead to some error, to some, as you say, mislabeling, and so on. And often we do not recognize this. In the past, we used our so-called reference collections. I mentioned very briefly in the beginning that we have, for example, in the case of cereals, we have always a sample of spikes and seeds. And if we find in a regeneration plot 2 different types of spikes, then we go back to our herbarium collection and check what was the original spike type. This was in the past. It's also nowadays we still use this possibility, but I think in the future one may have also some kind of a fingerprint for the different accessions where you can check after harvest whether it is still the same as it was. But you should keep in mind that most of these accessions are not homogeneous lines. They are land races. They are have a cross-pollinating species anyway. They are population. So this will be very, very difficult. But what we can do is to try to work as careful as possible. Okay.
Okay. Thank you, Andreas. I have a question for you.
Yes, please.
How many years are required to change germination percentage from a genotype? So if like I tested the germination for a genotype, like I found 100%.
Yes.
And then after 2 or 3 years, do you think that germination percentage will be affected if I stored the genotype at room temperature, for instance?
Yeah.
Or
it depends on the storage conditions, right? And it depends on the storage temperature and the moisture content of the seed. There is a so-called thumb rule of which was made or which tells us that if you either reduce the temperature by 5 degrees Celsius, or if you reduce the moisture content of the seed by 1%, you double the life of the seed. Yeah.
Okay.
So to keep it as dry and as cold as possible. But at room temperature, when you talk about barley or wheat, we have seen in, if you remember my slides, so 4 or 5 years, it's fine. But it must be dry, yeah?
Okay, thank you so much. We still have some questions, so I kindly ask the audience, our attendee, to send an email to Dr. Andreas Börner.
Yes,
if you go to the website, you will find the contacts of Dr. Andreas and the work of abstract. So please go ahead and send him an email about your questions. So we have to start with Dr. Ahmed Al-Quda. So could you please, Dr. Ahmed, share your slide?